The title of my talk is An Integrated Approach to Managing Immunogenicity Risk and Optimum Protein Design. We've had a few introductions from this topic from Laura in relation to allogenicity. We've been thinking quite a lot about vaccines this morning, and obviously a lot of it has been focused on cancer immunotherapy as well. But there's a lot more subject matter to cover today, so I wanted to start off just by kind of giving a bit of an introduction to the topic of immunogenicity, which will be very familiar to some, and to others probably this may not be such a kind of a core area of your focus. Now, essentially, immunogenicity, in this particular topic, I'm going to be focusing on thinking about unwanted immune responses, particularly to biological drugs, but not necessarily. It can also be consumer products, it could be sort of industrial enzymes, it can be food ingredients, such as we've heard about before. And essentially, all these types of therapies that are being developed can potentially have novel protein content. And in that case, if that therapeutic has been delivered to a patient or an individual, then a potential immune response can be mounted against it. And that can be a problem because ultimately, if you don't want to have an immune response against your biological drug, like an antibody, then the issue about generating the strong immune response can be that you get neutralization. And then the whole point of your therapy is impacted quite severely. So unwanted immunogenicity can result in treatment failure, and we don't want that. And so how do we stop it? Now, immunogenicity is really driven by a very complex set of factors, which there is really no one way and no one method that you can use to understand what's going on. The way that we kind of look at these things is that you can kind of split that down into both extrinsic and intrinsic factors. So extrinsic factors are such as the route of administration of the therapeutic, how often you're being exposed to it, whether it's a single bolus or maybe multiple exposures, which obviously increases the risk, a bit like a vaccination and generating an immune response. How much protein is being delivered, maybe even what the patient's immune system status is. Are they hyper-responsive or hypo-responsive? What about their HLA status? So the genetics of it really make a big difference as well. Then there are also intrinsic elements, such as the amino acid sequence, which we have some control over, but other things which we don't have so much control over, such as aggregation of the protein, which can also lead to strong, potentially unwanted immune responses. So if you're generating and developing a new protein therapeutic, you want to make sure that you have the lowest possible risk of an unwanted immune response against that drug. And so thinking about that at an earliest possible stage of design is really, really important, because this can make the difference between success and failure of your product. So this simple cartoon really is just a way of trying to sort of demonstrate the immune responses sort of approach to dealing with extrinsic protein. So here, a biological drug, this could be a therapeutic antibody, for example, is delivered to a patient and can be taken up by antigen-presenting cells. These APCs will then process and present peptides derived from that biological molecule in the context of MHC class II. So these peptides are then presented on the surface of MHC class II to T-cell receptors on CD4 helper T cells. If this specific interaction occurs and there's appropriate co-stimulation given by the APCs, then these T cells will become activated, they'll proliferate, they'll produce cytokines, and those cytokines will go on ultimately to help our B cells, which also recognize the same antigen, but obviously through surface-bound Ig, to become activated and develop into plasma cells. And these plasma cells then secrete antibodies against the very target that we're actually delivering to the patient. And these are referred to as anti-drug antibodies, or ADA. Now, anti-drug antibodies can come as neutralizing antibodies, non-neutralizing antibodies. There's all sorts of different aspects of them. But ultimately, they can bind to your target. That's what they do. And that can result in loss of efficacy of your drug, so altering the pharmacokinetics and pharmacodynamics. And in really serious cases, induce cross-reactive immune responses, actually against endogenous proteins. And that's really the most serious of situations where an otherwise an individual who may be treated for a particular disease is then generating an immune response against endogenous proteins which they rely on to survive. And there have been cases where immune responses that have been generated by biotherapeutics that are supposed to be helpful have actually resulted in death because of this very situation. So we don't want that, of course. So at ProImmune, we have a number of different assays to mitigate these risks. And the way that we do that is through a range of different tools, which I'm going to be describing today. The first sort of thing I want to introduce you to is our biobank. So we have access to PBMCs from healthy donors. And these PBMCs are all HLA typed, for MHC class II, so we know the full distribution of their HLA, DR, DP, and DQ alleles. And that's important because it means we can then select those donors for inclusion in our assays to understand the immune response in these healthy donors across a wide range of genetic allotypes. So these cells we then use in our functional dendritic cell T cell, antigen presentation assays, and just straight T cell assays. So you've already heard Margot speak this morning about ProPresent, and Laura as well about ProPresent, so I'm just going to give you a little bit more background on that and share some other data too. So ProPresent is a antigen presentation assay which is exquisitely sensitive and allowing you to sequence and identify the peptides that are naturally processed and presented in the context of MHC. Margot was showing you data from MHC class I presentation earlier with the glioblastoma and parvovirus case study. This data is really focusing on MHC class II. So it allows us to really understand which compartments or which elements of your drug are being processed and presented to the patient or the individual that's being exposed to it. It allows us to look at the impact of design variants, so maybe there have been amino acid point mutations introduced, maybe different formulations will alter how that protein is being presented and ultimately been recognized by the immune system. So it's a really great technology which marries up our own expertise of immunology and also the great advances in the past decade of proteomics, and bringing those together is really the massive advance that we now have. It's a really quick tool, so from a point of receipt of the proteins, we can then analyze these immune responses and report them back to you in just 4 to 6 weeks, depending on the scale of the project. And we have a lot of experience in a wide range of different types of molecules, mainly antibodies, but a lot of blood factors, gene therapy vectors, infectious disease, oncolytic viruses, and food proteins as well. And these are just some of the publications listed. So you've also seen this cartoon, so I won't spend much time on it, but basically protein is taken up by immature monocyte-derived dendritic cells. These DCs then are matured, they upregulate MHC class II. The protein is then process presented in the context of class II, and then we do an immunoprecipitation to pull down the MHC peptide complexes of interest. So we can focus on DR, DP, or DQ, or all three, or any combination of them, as well as MHC class I. But for the purposes of this, I'm just, again, focusing on class II. The peptides are then isolated from the groove of the MHC and then sequenced through tandem LC-MS analysis. So we can sequence all the peptides that are presented from the MHC molecules, and then we report to you all the peptides that are derived from your protein of interest, or mixture of proteins of interest, I should say. So the type of data that you get is really a really interesting insight into the repertoire of peptides presented from your drug. We have all the different donors and all the different HLA types of those donors listed on the left-hand columns. And as you can see for MHC class II, we get these nested sets of peptides. So peptides that will be overlapping by a number of different amino acids and a kind of core regions that are being presented. So you don't tend to see that the whole protein is being presented by the MHC. Of course, it's really key regions. And just because they're presented doesn't mean to say that they're actually functionally relevant. That's another question. This just tells us, are they there or not? And what we report is each peptide that's identified with an expect value, and an expect value is really a statistical measure of the identification. So how accurate is that measurement? And what we can do is really give very, very high certainty that that particular peptide is present. Not only an amino acid change, because that would really make a huge difference and it wouldn't be a significant response at all, but actually even subtle differences such as post-translational modifications can also be used in this to high significance. So you can identify peptides that have been oxidated, phosphorylated, and different kinds of PTMs as well. So to give you some examples, this is a case study looking at Vitreptocog alpha, which is an engineered factor 7A analog. It was developed by Novo Nordisk, and originally native factor 7A had been used for a very long time with no problems. The Vitreptocog alpha was sequence modified to increase its enzymatic activity. So they just introduced 3 point mutations, one C-terminal and one N-terminal and 2 C-terminal mutations. So on the face of it, pretty similar. Now, when it got to the phase 1, phase 2 trials, no problems, but actually when it got to the phase 3 and the numbers of patients became much more significant, it turned out that these anti-drug antibody inhibitors were observed in some patients. And for factor 7A, that's a really critical issue because it's the last line of defense. There's nothing else you can do. So you absolutely don't want to have these inhibitors at that stage. So late stage development was halted by Novo, and they really asked a retrospective question. How could we have done this better? Why did we not realise this beforehand? And a great paper was published between Novo Nordisk and the FDA. And this paper, which came out at the end of 2017, is actually really a good case study of understanding how these kinds of assays that I'm talking to you about really can make a big difference in understanding the potential risks with product development and how you can stop those happening. A lot of different assays were used in this paper, but one assay was that Novo Nordisk performed the ProPresent assay both with us, and they also did it blinded to us in parallel in-house. And so what you're looking at here is an analysis of recombinant factor 7A at the top and Bactreptocog alpha at the bottom. And their study looked at 17 donors, and we looked at 12 donors. They were completely independent. The methods were completely independent. But to cut a very long story short, we identified exactly the same region of peptide being presented in a high proportion of these donors. And when you look at these particular peptides that were identified, they are interesting right over that stretch of the two C-terminal mutations. So the N-terminal mutation wasn't a problem, but the two C-terminal mutations certainly were. And when you then synthesize those peptides and run them in a T-cell proliferation assay to measure the potency of those peptides to induce a T-cell response, you can see that the N-terminal mutation had no differences, but with the mutated sequences in the C-terminal, these both drove significant responses in healthy donors. They also went on to look at physical binding assays and HLA association, and really confirmed that these particular peptides, that the two mutations at the C-terminal were presented by HLA-DR and bound to these DR alleles with a really high affinity and resulted in T-cell activation. And the conclusions in the paper were that immunogenicity was the root cause failure of this particular drug, and that stratification of patient data by HLA type is essential when evaluating immunogenicity of any sequence-modified drugs. I mean, it's an obvious thing to state, but you'd be surprised how many people don't think about it. And also that a range of these different preclinical assessment tools, in this case, reflected the clinical outcome. And so really, we've got a lot to learn from it. I'm not going to do this slide justice with the author of the data in the room, but nevertheless, I will try and do my best. This is a paper, this is an abstract from a paper that, from a presentation that came out back in 2017, looking at Avastin, which is a B-domain deleted factor 8 protein. And this is comparing full-length recombinant factor 8 with the B-domain deleted Avastin product from CSL Behring and a plasma-derived factor 8 protein as well. And what you can see, this is a ProPresent data set looking at DR, DP, and DQ peptides. What you can see is there's a massive gap in the central region of DR, DP, and DQ derived peptides. Now that's not surprising for Avastin because that region does not exist. But for plasma-derived factor 8, you can see actually there's also a massive elimination of peptide presentation when you compare that to full-length recombinant factor 8, which is also commonly used by a number of different products, commercial products out there. And so the question that CSL wanted to address is really to understand the impact of the antigen presentation with their product and comparing that to full-length factor 8. This was done in the presence of von Willebrand factor, which binds to factor 8 with a really high affinity. And I'm excited to say, Mark just updated me that there's a paper coming out. It was accepted for publication just a couple of days ago in Blood Advances. I was going to say blood, that's fine. Blood Advances, where this is really a really nice set of data which I would recommend having a look at. And in actual fact, Zubin Sorna, who also was collaborating on this study, has given a talk at one of our previous meetings, and this is available on YouTube. So if you're interested in learning more, I'd recommend watching this clip. These are all linked through our website. But the great thing about this is that as well as looking at healthy donors, we were also able to look in a really unusual cases at patients who were haemophilia A patients. And getting blood from haemophilia A patients isn't an easy task, but nevertheless we managed to work through this collaboration to be able to do ProPresent on those samples, and we basically saw no differences between the healthy donors and the haemophilia A patients as well. So it's a really interesting study, and yeah, I recommend that to you to have a look at if you're interested in more detail. As well as factor replacement products, which of course immunogenicity is a really big issue, we've also performed this on multiple other types of proteins, including therapeutic antibodies. And this is again another video that if you're interested in looking at to identify the peptides that were presented from IPI and nivolumab from Merck as well. So we can have a look at that video too, but that's a really interesting case. There's so much to talk about, I can't fit it all in. Just because peptides are presented, I've mentioned that functional responses are also really the key thing to actually measuring if there's a relevant T-cell response. So to do that, we run our CFSE proliferation assays, which uses flow cytometry to assess the functional relevance of these individual peptides. And these assays essentially use peptides or PBMCs from multiple donors, and we're using peptides that are derived either from the ProPresent assay that have been identified from ProPresent that are being presented, or we can do overlapping peptides from a region of interest or the whole protein. So essentially we're labeling the PBMCs with CFSE, 7-day assay, you get proliferation, dye dilution, you can then measure exactly which cells are proliferated that are viable. And what you can then do, of course, is to identify hotspot regions. This is looking at an antibody CDR3 domain. So each of the different colours is a different donor that's responded. So you can see the kind of key hotspot areas. But not only that, we can combine this data with the ProPresent data. These are all overlapping peptides from the antibody. And this particular peptide here is one that's being presented very, very regularly and to a high level in pretty much most of the donors that we look at. So you can use those sorts of data sets to really see whether there's a functional response from those peptides that are being presented. What if you don't care about epitopes at all, and you just have a number of different proteins that candidates that you want to select to take forward in your therapeutics? So this is where the dendritic cell T-cell assays come in. So these are, again, monocyte-derived dendritic cells we generate from healthy donors. We load them with your proteins of interest. We then mature them, and we do a co-culture with autologous PBMCs, which are CFSE labeled. So here, we're measuring T-cell proliferation as a direct result of that protein being processed and presented by the DCs. We look at a panel of donors, normally 40 or 50 donors, that are all, again, HLA typed. And the nice thing here is there's no sort of really a direct exposure of the test article, because that's washed out. And it's applicable to assess issues of post-translational modification, also aggregation effects, host cell proteins, also other excipients can all be used to good effect in this assay. So the outcome of the assay, we also have assay controls, which are just really to show that the donors are responding really well, which with KLH and PPD they of course do. But these are therapeutic proteins, in this case a study that was published by Elasmogen, working on shark VNAR domains. And you can see here, these kinds of levels are very typical for most therapeutic proteins that we get when we're looking at a large panel of donors. But the nice thing about these DC-T cell assays is that we take into account both the percentage of donors that are responding significantly, but also the strength of that response. So because you have a really wide dynamic range of these assays of the dendritic cells, you could potentially have just 1 or 2 donors responding that are really, really strong responders, but that means something a little bit different to lots and lots of donors responding but at very low levels. So the granularity of that data is something that's very easily achieved with these sorts of assays. Again, another case study from TIVA looking at the impact of host cell protein contamination. This was a study where different levels of HCP were spiked in, so you can see that as you do that, you get obviously a much stronger response. This was done in a blinded assay, so we weren't aware of exactly what the test articles were. But again, this was another interesting way of being able to use these assays to address these kinds of questions. Talking now about Cytokine Storm, which was mentioned by Lucy earlier on as part of the strategy at Immunocore for looking at one of the risk factors associated with these TCRs. Cytokine Storm is obviously a pretty hot topic. You need to understand exactly what the potential risk of innate and rapid immune response is if you deliver a therapeutic. Cytokine Storm is really where you get, as the name suggests, a lot of cytokines produced. Typically IL-6, IL-8 are some of the key cytokines associated with this upon rapid production upon delivery of your therapeutic. There is guidance from both European and US regulators that say that data from animal studies should be supplemented by in vitro assessment of cellular activation, including proliferation and cytokine release in human whole blood or PBMCs, and that you need to look at a wide panel of cytokines. So what we do in ProImmune is we use a whole blood undiluted assay, where the blood is taken from healthy donors, healthy volunteers that are recruited, and that blood is then drawn. These are healthy donors so that they have no symptoms of anything, they're feeling healthy, they're adults, they're not taking any immunosuppressants or any paracetamol or any ibuprofen, and that blood is then taken straight up to the lab within just a couple of hours, incubated at a range of concentrations with a test article. We then, after an overnight incubation, isolate the plasma and then measure the cytokines produced in a multiplex format. So it's a very nice way of being able to, as close as possible, match exactly what's happening in patients. So we typically also run this assay, or not always, against a couple of comparators. So the assay controls are always included. We have PBS and staph enterotoxin B as positive controls. But we can also compare this against Erbitux, which is cetuximab, a very low response comparator, and then alemtuzumab, anti-CD52, which is right up there. And pretty much most test articles will always be in between that range. So the data is read out by all these different diamonds of different donors, and as we increase the drug concentration logarithmically at the top, you can then measure the, in this case, IL-6, the amount of cytokine produced in picograms per mil using a microarray format approach that we use against a standard curve. So what you can see is that there are sort of high-risk proteins, such as our high-response comparator Campath, which is known to induce strong cytokine storm. And then we have Erbitux, which is our low-response comparator down here. And then you typically will have your test articles like Remicade, Herceptin, Vectibix, whatever that might be. Normally they're against the same target, but they're just a range of different therapeutics. And this molecule will then, as you increase the concentration, more and more donors will then become positive. So you can determine where that particular test article lies in the range of different entities. Interestingly, if you aggregate Remicade, you can see the effect of that, presumably upon engagement with TLRs on the surface of the cells in the whole blood. That's obviously something which is really driving the innate immune response. So from this, we can understand exactly the response of your own test article and where that lies in that particular range. So it's a hazard identification assay, a very simple but a simple system in the sense of its design, but it really provides a lot of information in relation to the risk of your own test article and how that fits in against others as well. And the nice thing about it is because we do the whole thing for you, this is usually delivered in just a few weeks, including all the donor recruitment. So we've got a very efficient way of getting all this sorted out. The last thing I want to touch on in the last couple of minutes is quite a different kind of service. You may have mentioned it, seen it on the front there, MutaMap. So this is a protein engineering tool which we have at ProImmune, a high-throughput mutational protein activity map, allowing you to really understand in a very high-throughput way the effect of introducing point mutations on activity or affinity of, for example, a monoclonal antibody, really helping you to make the best protein engineering decisions that you can in a really quick way. Of course, there are many ways of doing this, and a lot of people will use multiple different approaches, affinity maturation, for example, to identify the most optimum clone. But the problem with these sorts of approaches is that they can take quite a long time, and it still leaves questions at the end of the process, for example, that how that ties in with immunogenicity. That can be one thing. I'll mention some other in a moment. So the way that we do this is we actually explore the effect of substituting each and every amino acid in a particular region of your protein with all the different amino acid substitutions. So typically you'd obviously focus in the CDR regions of your antibody. So we take the parental sequence, we then perform site-directed mutagenesis to introduce that one-point mutation. We then express that protein through in vitro translation. And then we don't use surface plasmon resonance or Biocore. We use high-throughput solution equilibrium titration assay, which is an old-school method, but incredibly powerful, particularly great for looking at high-affinity interactions. So it allows us to really understand the measurement and the affinity measurements of high-affinity interactions, where, as with SPR, you have a problem because the dissociation of the antibody to the ligand takes a long time to come off. Whereas when you're working with more of a solution equilibrium constant, you can actually get that measurement in a very, very accurate way. So this allows us to fully characterize hundreds of mutations in just a number of weeks. So to give you an example, I'm looking here at comparing Avastin to Lucentis. These are both anti-VEGF antibodies, and Lucentis was a modification including 5 amino acid substitutions from the parental Avastin sequence. And this curve here just shows you that using the solution equilibrium titration assay, the improvement in affinity of Lucentis over Avastin. So you can see here the KDs reduced from 16 to 4.2 with Lucentis. Now, what I want to show you now is we take the Avastin parental sequence, we then generate a heat map of the impact of every single one of those point mutations on the parental sequence. So what you're looking at here is the Avastin sequence down the y-axis, and then along the x-axis are all the different amino acid variants. As it's a heat map, it's fairly self-explanatory. Red is really poor affinity, or it doesn't express. Dark orange is poor affinity. Yellow is essentially equivalent affinity, and green is better. Oh, that's my time up. I have to judge myself, you see. So the thing to point out here is that you can see there are particular regions which really are not permissive at all to engineer, such as this tyrosine here. The only option is actually a tyrosine. Everything else is worse. So this kind of information allows you to see which regions you can tinker with, which ones you can't. And because it's actually expressing the protein through in vitro translation, there isn't the question of, oh, well, can we express this or can we not express this? This is really, really a functional measurement of the affinity. So there are other regions which, of course, are quite permissive and actually allow a number of different changes to be made. If we focus at this particular serine residue here, I'm just going to show you a bit more of the raw data. You can see that compared to the wild type, we have one really improved affinity change with the valine, and then we have a number of sort of neutral affinity changes, and then a number of reduced affinity changes. Interestingly, this serine threonine mutation is actually the mutation that was incorporated into Lucentis. For some reason, they didn't take that one. There must have been another reason behind that. But the key point to take home from this assay is that it's really, really quick at being able to identify a huge number of changes. I mean, we're talking about hundreds or even thousands of mutations. And this information will allow you to have choices. It allows you, in the sort of fast-paced arena of making the right choices quickly in pharmaceutical development, it allows you to make those right choices with a number of different characteristics in mind, developability characteristics in mind. And that's very, very important. So it allows you to quantitatively determine the impact of those different changes to help you make the right choices. So you can obviously cherry pick the right mutations, but you can also use it for other reasons. You might want to get an improved cross-reactivity with the different species in the preclinical development. You might be wanting to improve the stability or other manufacturing elements. You might be wanting to extending the half-life, or of course it generates new composition of matter intellectual property as well. So all of this is a very, very quick method of being able to get to the heart of the matter. So just to summarize, managing drug immunogenicity is really, really key to some of these questions. If you don't make the right choices, you'll be stuck with a very expensive mistake. And ProImmune, I hope we've demonstrated, is really the expert go-to company to work with for some of these key questions. And because of the breadth of experience that we've got, we pretty much work with most entities, if not in the past, then currently. So thank you very much for your attention, and I welcome any questions.